Thank you very much, Andres. Please,
Good.
the floor is yours. So go ahead. Thank you very much.
Great. So I assume I have 45 minutes or something?
Yeah,
Good.
45 minutes.
Okay, good. So thank you for the invitation, first of all. And so today what I want to talk about is about a framework that I've been using for a while, for the last 7 years, to study aspects of perception and also a bit of predictions in the brain. And the main message here is just to show you how you can use that is based on to to basically explain or try to understand certain phenomena that is not as obvious as you would infer out using other metrics like spectral measures or time frequency charts, etc. So I'm going to explain what I mean by that. But the main motivation here is that the brain, as we know, is a complex system, and perception in particular is not something that it's just the result of bottom-up processing, but it's kind of the interface between the information that comes from our senses and the information that we have stored somewhere, like predictions or attention or any kind of top-down effects. Well, the thing now about perception is that perception is more like an active process that incorporates feed-forward activity, information from the senses, and also top-down activity coming from predictions or attention. So, and this is based on previous work of other people, in particular from Walter Freeman and Francisco Varela, who basically believed that perception was an active process rather than just a passive process of information. So, and a second aspect is not about perception itself, but it's about the underlying neural dynamics. And in this paper a couple of months ago, what we were trying to say or try to explain was that if you care about studying perception, you need to distinguish between two types of processes. One is the activity that happens in the brain that relates to the transmission of information from one area to another one. And another process is the aspect of the integration. And the point that I want to make here is that you can distinguish clearly these two types of process based on different types of metrics in the brain, ways of computing activity in the brain. And it's long to explain, but in principle, what we want to say is that if we care about the aspect of the information processing that is important for perception that relates to the integration, so to the ability to different areas to actually process information together, we need to care about information theory metrics and not much about connectivity tools that rely on spectral decomposition. And the reason is that these information theory metrics are capable of capturing nonlinear interactions. And we believe that these kind of nonlinear interactions in the brain are the ones that are responsible for the actual information processing. So during this talk, what I'm going to do is try to give you 3 examples of how you can use information theory to separate different or to study different perceptual processes. And also I'm going to make the contrast between those metrics that comes from the world of information theory against metrics that we are traditionally using in the EEG, MEG, eco community, like phase coherence and others that are derived from the spectrum. So I'm going to try to make the contrast between the two. And for that, I'm going to explain first one study that we ran a few years ago, and is based on perceptual ambiguity. And of course, you know these kind of illusions, but the idea here is that if we care about perception, we need to care a bit more about the internal aspect of it. And an obvious thing about perception is that it varies depending on the interpretation that we assign to different stimuli. So for instance, here we can see this classic example where you can actually interpret this image as an old lady or as a young lady, depending on your perspective. Although the information that comes to your senses, to your eyes, is exactly the same, you can perceive 2 different interpretations. So what we did was basically to incorporate these concepts into the idea that perception is not something that is unitary. So, I mean, it's something that is unitary, but it's also and a phenomena that is also differentiated. So what we wanted to study was aspects of perception that were related to two phenomenological properties. One is the feature of perception that is integrated. So basically, we experience our environment as a unitary whole, as a unitary thing, but at the same time, we can make distinctions. So there are theories in the field of consciousness and in the field of perception that basically claim that these two aspects, the integration and the differentiation, go along in every kind of experience. So what we wanted to do was trying to dissociate these two experiential phenomenological aspects of perception and try to map them into the brain. And our hypothesis was very, very simple. And what we wanted to test was the idea that when you experience one of these illusions in a more integrated way, you should see more integrated activity in the brain. So basically, the perception integration should go along with the neural or the brain integration. And when you experience these illusions in a more differentiated way, so for instance, here when you see two faces instead of one base, we could say that there are two objects rather than one. This perception would be more differentiated than when you see a single object. Well, that aspect of the perception should go along as well with more differentiation in the brain. So the idea was that we wanted to map these two properties of perception, which are integration and differentiation, to map them with some corresponding metrics in the brain, so neural integration or neural differentiation. And we did that not by using a visual stimuli, but we did it by using an auditory stimuli. And the idea is that you can generate an illusion, a bistable percept, that has these two properties of integration and differentiation by playing two tones together over and over again. So it basically sounds like bitty beep, bitty beep, bitty beep, bitty beep, bitty beep, bitty beep, so on and so forth. And after a minute or so, what you start hearing in this stream of sounds is either you experience these tones as an integrated percept that sounds like a galloping rhythm, taka-tak, 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 or you can also experience this as two separate streams where one of the tones is in the foreground and the other tone is in the background. I cannot repeat that, but you have to believe me that when you hear this, you either experience these tones as one object, one auditory object, or as two auditory objects. So basically, the illusion has a percept that is more integrated than the other and a percept that is more differentiated than the other, because you jump from one object to two objects to one object and to two objects, etc. So that was our manipulation of integration and differentiation in stimuli. And then when we asked the participant was to press a button every time that they experience the change from integrated to differentiated, and another button when they experience the opposite, so from differentiated to integrated. And we measured the brain activity 2 seconds, 2.5 seconds before the button press to map, to investigate whether this perceptual integration would go along with some kind of metric of neural integration. And our metric of neural integration was based on information theory. The metric that we use is called WSMI, or Weighted Symbolic Mutual Information. In short, they call it information sharing, but the main point of the metric is that unlike metrics that are based on spectral decomposition like phase coherence or phase locking value or Ranger causality or any other metric that is based on spectrum, this metric, what it does is that it computes the shared entropy between signals. And the ability of this metric in particular is to capture interactions that are nonlinear, so interactions that are not necessarily proportional to each other. And it does that by taking the signal and transforming it into little symbols that have a specific length. And then if you have a signal X and a signal Y, you can actually map all the little symbols in one signal and the other. And what you do is basically you calculate the probability of these symbols to co-occur simultaneously. Like for instance, this green one against this blue one, or this orange against this young one. And you compute the probability over time of these symbols to co-occur. So the idea here, and this is the way how I explain this all the time, is that instead of having a pattern that is the same in the two signals, and you call that connectivity between areas, for instance, when you have the same phase, What you're actually computing here is the co-occurrence of different patterns. So in some way, this measure of connectivity is like dancing together. And the metaphor is like, okay, if we dance tango, then we are coordinated between each other, me and my partner, but we don't need to be doing the same thing at the same time all the time. We can be doing different things. So for instance, one dancer can be in this position and the other one is the other position, and so on and so forth. And we cannot deny the fact that these two dancers are actually coordinated. So we can have coordination in the brain, communication in the brain if you want, integration in the brain, when two signals are doing different things, right? So this metric of integration is actually a very good candidate for capturing this endogenous activity for a reason that I'm going to give you in a few more minutes. But this was our measure of integration, this nonlinear information sharing. And also we needed a measure of differentiation because we wanted to map the properties of perception with the properties of the brain. And the measure that we used was this Lempel-Zip complexity that I think most of you already know. But the main idea is that if you have a signal, like an EEG signal or any other signal, and you binarize the signal in a way, for instance, you take the mean of the signal and you put a 1 over the mean and a 0 over the mean, you can binarize this signal into zeros and ones, and then you can try to compress it exactly like you compress a zip file. And the idea here is that if a signal is fully compressible, so if you really, really compress it, it means that the patterns of information that contains are very repetitive, so are not very differentiated. So if a signal is fully compressible, it means that the signal is not very differentiated. It has very repetitive things inside. But if the signal is not very compressible, then you can claim that the patterns of information inside the signal are not repetitive. So you say that the signal is less differentiated in terms of information. So you can have an index of neural differentiation based on the amount of compression that you have in the signal. So going to the experiment, here are basically the results. Just focus on this side of the screen for now, please. So this is the metric of nonlinear integration that I showed you briefly before. This is the moment where participants press the button when the stimuli changes from 2 objects to 1. And this is the moment in the EEG, 2.5 seconds before the response. These are 33 participants, and this is the nonlinear integration between frontal sensors and more parietal sensors. And what you see here is that before the participant pressed the button telling me that they're feeling the integrated percept, the measure of integration goes up. So, when they are reporting a more integrated percept, the integration between frontal and parietal goes up. Now, if you see what happens with the neural differentiation metric, we predicted that differentiation should go along with perceptual differentiation. So here, when the participant experienced the percept as a differentiated one, so 2 objects instead of 1, now this measure of compression, that is a measure of neural amount of information differentiation in the brain, this one goes up. So the conclusion of these 2 images is that, actually, of these 2 figures, is that perceptual integration goes along with neural integration, and perceptual differentiation goes along with neural differentiation. So that the metric and the perceptual experience go hand in hand. Now, you can say, well, but how do you know that this is only valid for bistable stimuli? How do you know that this effect is not present when you have other type of stimuli? So we had a control where we actually changed the sound. Remember that the sound here doesn't change. It's the 2 tones all the time. The only thing that changes is your interpretation. But if you actually change the sound to sound like 1 object or 2, the metric doesn't signal the change. The integration signal nor the differentiation signal. So it's only when the change is endogenous, it's only when the change is internal, like in the case of the bistable stimuli, that these 2 information theory metrics signal the 2 types of percepts. And we found exactly the same example, or the same effect, when we computed the same metrics, but now in an intracranial patient with electrodes in the superior parietal lobe and in the mid-frontal gyrus. These patients are patients that are resilient. No, they're basically they cannot resist. They're not good with the drugs. So basically, they don't get better with the drugs. So they got into surgery, and then when they're into surgery, we can run this experiment. But the main idea here is that you have exactly the same effect. So integration goes up when they say that they're experiencing the integrated percept, and differentiation goes up when they say that they're experiencing the differentiated percept, and the control doesn't show any effect. Now, you might be asking yourself, so, but what happens with the normal metrics, or with the spectral metrics like phase coherence or PLV, and you know these metrics because they're very popular, and the main idea here is that you have two electrodes and you compute the phase of a signal in a specific frequency. You can say that these two signals are well synchronized if the difference in phase is consistent across trials, or they are not very synchronized if this phase difference across trials is all scattered. And this is, I call it like, my example is like jumping together. So basically when we jump together, even if we jump with a little delay, we're actually doing the same thing at the same time, or doing the same thing with a little delay. And we consider that a candidate for integration. But the thing is, when we computed these signals in exactly the same trials, in exactly the same participants, we couldn't find any difference between the two types of percepts. So it's only the information theory metrics, the ones that capture the difference in perception and this bistable stimuli. It's not the phase in the same range of frequency that we observed the effect in the information theory metrics. So there is something about these information theory metrics that capture these effects, and we believe that is because of the fact they're based. These metrics can actually capture nonlinear effects. But I'm gonna get into that in 3 seconds. Now I'm gonna show you a fish, and then you're gonna say why Andres is showing us a fish. Well, for a good reason. So we have these colleagues in Valparaiso when I did my undergrad, and then my colleague said, hey, we have these beautiful signals coming from the olfactory bulb and other pieces of the brain that we see this beautiful oscillation when you present to the fish different odorants, so different smells, and they have a nice peak at 7 hertz. So I'm pretty sure, my colleague said, Oliver, that the distinction between the types of odorants has to be in the phase coherence between the signals, because if you have a beautiful oscillation, of course the synchronization between the signals must be the responsible, or at least it should correlate with the differences in odorants. So we tested again the two metrics, the spectral metric based on phase and the information theory metrics based on chair entropy. And interestingly, while the phase coherence signal only distinguished the 3 types of odorants against the control, not between each other, the information theory metric could actually separate the 3 odorants between them and all the odorants against the control. So you get much better discriminability using these information theory metrics as compared to using these spectral ones. So again, another example where these information theory metrics are more powerful to distinguish between types of percepts, in this case, in a fish. So now you would be asking yourself, okay, but why? So we try to test this idea that because this information theory can in principle capture nonlinearities in your signal, then they should be more sensible to nonlinear interactions across the brain. So we ran a study where we just simulated signals. So we simulated EEG signals in the parietal and frontal cortex. And then what we actually modulated was the type of interaction between these areas. So we made it like extremely linear and proportional, or we played with different amounts of nonlinearities. So basically, we simulated signals that were extremely, the relationship between them was proportional to each other, or it was less and less and less proportional. So we tried different maps, different types of nonlinearities. And then we tested our information theory metric against the spectral metric of connectivity. And the prediction was that this WSMI, because we believe that captures nonlinearities, it should be very good at capturing nonlinearities when our simulations are highly nonlinear. and the linear one should be very good at capturing linear ones. That's exactly what we found. So basically, the more nonlinear the relationship between the signals is, the better the WSMI captures those nonlinearities. So even if the relationship between your signals is not obvious, but we know that it's present because we simulated it, this mutual information metrics can actually capture it. So we believe that that's why we could find those differences before. Okay, so now I want to give you another example, also on bistability, but in a visual case. And we repeated the same type of analysis or the same type of approach, but using a visual stimuli for a good reason. And the reason is that in my field, I study perception and consciousness, people say that when you find a neural marker of a perceptual state, that might be always confounded by the fact that you need to ask the participants to press buttons. So you're always confounding the perceptual effect with what they call the post-perceptual effects. So the act of, I don't know, making a decision, thinking about which button are you going to press, the motor activity itself. So that those processes that are not perceptual in themselves, they can be obscured or they can be confounded by these post-perceptual processes. So we need to find a way to separate what is perception from what is post-perceptual. And what we did, we come out with this very simple stimuli that is also bistable. But the interesting thing was, if you look at this side of the screen, is that now you have these gray things that you see here. They're always moving on the screen, but you can perceive them as moving together upwards. So again, one object, we call this again the integrated one. Or you can see these two gray things moving apart from each other, like two objects. So you can either perceive, again, one integrated object or two differentiated objects, but now in the visual domain, and you can jump spontaneously, your perception jumps from one interpretation to the other, right? But we added critically another condition in which the participants were seeing the same illusion, but they were not pressing buttons. So we had a condition when they were actually reporting the changes using button presses, and another condition when they were not pressing any button. And we were inferring which state they were actually experiencing by co-registering the eye movements with an eye tracker. So we used a technique based on some involuntary movements of the eyes called optokinetic nystagmus, or OKN, that allowed us to decode whether participants were seeing one or the other based on these eye movements. So basically, if you train a classifier with the eye movements to detect whether they're going to press one or the other button, you have over 85% accuracy in discriminating whether the button is going to be differentiated or integrated based on the eye movements. So then we can use exactly the same decoder to infer the states, but in the condition when there are no movements. So it's like a bit of a mind reading in a very basic way. But in this no report condition, you can tell whether they are seeing integrated or differentiated without the button presses. And again, we were interested in the dynamics, and in particular in the information dynamics, because as I showed you before, the phase coherence doesn't show much. And we ran a very similar analysis, and the idea was exactly the same. We were expecting more integration in the neural data when participants were reporting, or even when they were not reporting the integrated percept. And we were expecting more differentiation when they were reporting and no reporting the differentiated percept. The only difference is that instead of the previous metric, we used a measure of information that is directional, so we can actually tell now whether the information flows from parietal to frontal electrodes or from frontal to parietal, based on the idea that these effects that are endogenous should be more related to top-down effects. So we were expecting the flow of information from frontal to parietal, but that was the only difference. The measure of differentiation was exactly the same as before. And so let me show you first what happened with the report condition, so where they were actually pressing buttons. And long story short, what you see here in the upper row is 0 is the moment when they press the button when they experience the integrated percept, and blue is when they experience the differentiated percept, so one object or two objects. And when you contrast the two, you see that around 1 second before they press the button, you see an increase in transfer entropy between front and back. So directional information flowing from frontal to parietal electrodes in the integrated case against the differentiated case. So again, integration, higher integration in the brain goes along with perception integration. But now we know that the effect is top-down. So it goes like from front to parietal. It doesn't happen, or we saw no effects when we tested the contrary. So information flowing from back to front. So this is consistent with the previous study that I showed you in the auditory domain. And then if you want, the interaction is there and blah, blah, blah. And a similar thing was observed for the differentiated metric. So this is the 0 is where they press the button. And then you see that the differentiation, so basically this measure of compressibility, goes up before they signal the differentiated percept. And it's an effect that happens in the front and not in the back. So again, perceptual integration goes along with neural integration, and perceptual differentiation goes along with neural differentiation. So you would say, well, okay, so what? Well, the interesting thing happened when we tested the no report condition, right? Because again, remember that what we're trying to do is to separate what is perceptual from what is not. So we don't want responses anymore. We want to infer the percepts based on eye movements. So no report, at least not explicit report. And the interesting thing was that we observed exactly the same direction of the effect. So higher information flow from front to back in the integrated case against the differentiated case, and we didn't observe it in the opposite direction. So again, higher integration in the integrated case when we infer the states based on the eye movement. So here there are no button presses. But the interesting thing is that the measure of differentiation didn't show any difference between the two types of percepts. So when we contrasted now the differentiation metric between the two types of percept, we couldn't find any difference in the front nor in the back. And how are we interpreting this? Well, one way of interpreting this, which is the one version that I like, is that the neural marker that is more or is more consistent with perception is this transfer entropy metric. So the measure of information that I show you that computes integration between areas is the one that is more consistent with perception, I said, because you have it when you're reporting, but you also observe it when you're not reporting. So even in the absence of explicit report, you still see the marker. But this measure of the information differentiation apparently maybe has to do only with the aspect of post-perception, because we didn't observe it in the case where we only infer the states based on eye movements. We only saw it when people were pressing buttons. So maybe it's only related to post-perceptual processing. So again, you can use this information theory metrics to disentangle, in this case, what is perception from what is post-perception. And now in the part 3, I'm gonna get a bit more I don't know, probably this is more new for all of you, I hope. But I'm gonna bring a very simple case of auditory predictions. And this is based on a preprint that we have out with Frank, Martin Vink, and all other people around. And this is to illustrate another type of metrics that are very, very useful. And I think they're gonna become more and more useful as time passes, that are based on not only capturing whether two areas or two regions are correlated to each other, either by sharing information or being in phase, but more importantly, these measures can actually capture the type of information that is being processed, right? And our question here was whether these effects of prediction errors, basically the brain is always trying to predict what's happened next, and you have ways of manipulating that by playing tones and then having an oddball in between, and then seeing what happens with different brain signals and how these different areas communicate information. But what we were testing here was the idea that these interactions were not just redundant, informationally speaking, but they were synergistic, informationally speaking. And let me explain what I mean by that. So again, we use information theory tools, but in this case, we use metrics of redundancy and synergy. And the idea here is very simple. So imagine that you have two signals. It could be like a signal recorded in V1 and a signal recorded in the frontal areas. And then if you have a difference between two types of stimuli, let's say two types of visual stimuli, or in this case, two types of different tones, you can compute how much information is being processed in two ways. You can either ask how much of the information is the same. So we call that redundant information. So the two areas are actually on a trial by trial basis computing exactly the same information. Or you can have what is called synergy. And synergy is a very interesting phenomenon because even though the two signals are actually communicated or they are actually integrated, they are not talking about the same thing, they complement each other. So the idea here is that this synergy is the complementary or extra information that you get when you know the two signals together. And it's this basic idea that the total is more than the sum of the parts. This is what synergy means. It means the extra juice, if you want, in information in bits that you get by combining signals together. That extra information that you get when you take two signals together that you don't get when you look at them separately. So this synergy is interesting because it has been related to flexible coding in the brain. And it also speaks a bit about these nonlinear effects at a distance in the brain. So in short, we had a very basic tasks. These tasks are the famous like mismatch negativity ones. This is not in humans, this is in marmosets. So we're dealing now with intracortical recordings here. But I'm gonna explain the results of the very basic task. So basically you have repeated tones, beep, 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 boop, 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 boop. So basically you have repetitive tones, and the idea is that the brain gets used to the same tone until there is something new coming. And the idea is that if you take those two signals, you can actually separate what is an echo of the spikes in the LFPs that is called broadband activity, or you can also compute the ERPs between your signals. And the idea is that your deviant, so basically the tone that deviates from the standard ones, will also show you a higher amplitude in the ERPs and also will show you a higher broadband than the standard tones. The broadband, again, is like an echo of the spikes that you can extract from the echo activity. And then our question, our hypothesis was that this prediction error, so basically this effect that you see here between standard and deviants, would be mainly be reflected in the synergistic information rather than in the redundant information. And we also had a model that I'm not sure if I'm gonna, I think I still have a bit of time. We have a model to try to understand what this synergy means. So what kind of architecture in the brain is more suitable for generating this complementary or these synergistic effects. And we had a model that I'm gonna show at the end. But for now, here are the three monkeys that we tested, marmosets. So here what you see are all the electrodes that show a difference between the standard and the deviant tones. You see that the ERPs are much widespread than the effects of the broadband. The broadband is a very local thing, is again a reflection of spikes. And then you can measure the difference between the two and you can calculate how big these effects are using, again, mutual information. But this is just to illustrate the fact that you can use information theory to compute as well effect sizes between your signals. And you can have a normalized or a standardized unit that is in bits. So basically you can compare now every contrast in the same scale of bits, right? So basically the effects are very distributed. They are like all over the cortex. And this is the main point here. So this is the measure of redundancy and synergy. And I'm gonna make this bigger so you'll see it. This is the case when you see an effect within an electrode. So if I take one electrode in the temporal cortex, and on a trial by trial basis, I compute how this information across time is related to each other, I can see two things. I can see effects that are in red, and I can see effects that are in blue. Everything that you see in red are values of the information that are redundant across each other. So basically, if you combine all of these points together, the information about the difference between a standard tone and a deviant tone is redundant across trials in this peak. That's why you see red there. So all the time points against all the contiguous time points in the same signal are redundant between each other. But you can also see synergy, and synergy is really cool, and it's represented by these negative values that are in blue. What this basically means is that if you combine the time points of the peak against the time points of the tail of the same signal, when you combine these points, you get more information by combining them than by studying them separately. So there is some kind of, again, non-additive or complementary or synergistic effect when you take together these points and you take together with these points. And again, this information is about the difference between the standard tones and a deviant tone. So in other words, you're better at telling whether the tone is a standard or a deviant when you combine the information in these two parts of the signal. That's what synergy means, right? And here what I'm showing you is just the redundancy and the synergy separated. And these are the number of pairs, or the number in this case of monkeys that show the same effect. So that's what synergy means intuitively. You gain better predictability, or you can distinguish better your stimuli by knowing two parts of the signal together than in isolation. Now, the important thing I would say is that we observe a lot of these synergistic effects either in the temporal electrodes, but also in the frontal electrodes. And they were mainly in the earpiece. There was some of it in the broadband, but they were mainly in the earpiece. Now, the main point of the paper was to show that this synergy or these complementary effects can happen between distant areas. And this is what I'm showing you here. So if you take the temporal electrodes and you combine them against what happens in the front, what you can see is that there is a lot of synergy. This gray scale basically shows you the number of electrode pairs that show an effect. Here, all the effects, redundant and synergistic, and I here just separated what the effects were redundant and when the effects were synergistic. Part of the synergy was also observed in the other neural marker, in the broadband response. So the message that I'm trying to convey with this slide is to show you that when you try to understand the information relationship between areas that are far away, that are distant between each other, sometimes you don't see only redundancy. So these two areas are not just processing the same information about the stimuli difference. Sometimes you see synergy. And what synergy means is that, again, you gain more information, or you can predict better the class of the stimuli, whether it's a standard or a deviant, by combining these two signals together, by combining what happens in the temporal electrodes with what happens in the frontal electrode. So these prediction error effects are in some way synergistic, and they're not just redundant. So this temporal synergy was observed across experiments and marmosets, and this long distance synergy was also observed between across experiments and marmosets. But you might be asking yourself a lot of questions, but one of them is, okay, what does this synergy mean? How can we generate these superadditive effects? And we have this model that what we did was basically to reproduce the anatomy of 6 different areas of the marmoset brain. And we played with the amount of connections or with the type of connection. So basically what we did, this is a neural network that you can simulate. They're called brain-constrained neural networks because basically they are neural networks that are connected in a way that resembles the brain that you want to simulate. In this case, the brain of a marmoset. And we tried 2 architectures. One architecture that is only feed forward. So basically the information flows from auditory areas to the front. So it's just unidirectional. And another network that has feedback. So On top of the feed forward, you have connections that are jumping and their feedback. Our idea of synergy, and coming back to the first and second study, is that it has to reflect some kind of nonlinear effect. So it has to reflect some kind of recurrency in the network. You cannot have synergy if you don't have recurrency. That was like the basic idea. By recurrency, I mean information that in some way, it's also feedback. It goes from areas that are superior to areas that are inferior in the hierarchy. So we were expecting to see the synergy in this architecture, but not in this architecture. That's the idea. And I'm going to show you the ultimate result. So basically, if you want to read the paper, it's longer than that. But first of all, we could simulate the effect that we observed in these prediction errors. And then what we observed in the fully connected network, so in the network that has feedback or recurrent activity, is that we observe redundancy between these areas, between temporal and frontal. And we also observe synergy. So there are complementary effects between these areas when the network is connected with feed forward and feedback physical connections. But the interesting thing was this. The moment that you cut the feedback connections, so the moment that you cut the recurrent activity, and you have only a network that is feed forward, you still see the redundancy, but you stop seeing the synergy. So in some way, these synergistic effects, as we thought, are the consequence, well, at least in this simulation, of this feed forward or recurrent connection, which makes sense because we think that this, again, this information theory metrics are the reflection of some kind of nonlinear processing, and that nonlinear processing can come in the form or in the flavor of recurrent connections, right? So to conclude, basically we observed synergistic interactions in these ERPs and in this broadband. They were distributed between areas, and we also show that this synergy probably emerges out of the feedback interactions in a network when we are capable of replicating the effect and simulating the redundancy and the synergy. So to conclude, what I want to say is that, or I want to motivate you to start thinking about your experiments and to start thinking about whatever question you have in mind, but maybe using an information theory framework, because I've tried to show you now, these effects are much more robust to extract from the data. Information theory allows you to put all your contrast into the same scales because everything is in bits. So basically you have entropy explained, which is like a information theory way of saying variance explained. You have not only the connectivity between areas, but also you can have the flavor of that interaction. You can know whether your areas are computing redundant information or synergistic information. And these metrics are by all means more sensitive than metrics derived from the spectrum. And we believe that they are more theoretically motivated because they capture nonlinear effects. And if I can say that, I think that every important or relevant thing in biology has to have some kind of nonlinear component. Otherwise, we wouldn't exist as a species. So having said that, I want to thank everyone, all the collaborators of all the studies, of course, Boris and Chiara in Chile, and thank you very much for listening.